Welcome back to the Felix Factor, with Felix Heron reporting. This week, we're talking about cells. Not just cells, but what goes on inside each individual cell in your body. We're talking about cells motion. So, most people know about things going on inside your cell. All the different organelles processing proteins, making DNA, moving stuff around, all that nice stuff that we're all about to learn about. However, some people overlook the fact that lots of cells move around themselves, like a person moves, except a cell. The way they do this tends to be using flagella, which are little tails on the end of cells that can beat, and kind of like your feet in the water when you swim, they propel the cell through interstitial fluid or through blood or through any other fluid in which the cell is. So how would we know about all the different happenings inside cells and even outside of cells if we didn't have microscopes? Microscopes. There are many different kinds of these. However, two main classifications. First, light microscopes, which use photons to bounce off through the specimen so you can see it with your eye, or electron microscopes, which use beams of electrons, which then get converted using computers into images. What you see here is a scanning electron microscope. What it does is it shoots a beam of electrons at a metal-coated substance, which then emits more electrons, which can be captured by a computer, which can then produce an image based on what it receives. What you see here is a transition electron microscope, abbreviated TEM. It works very similarly to a light microscope in that it sends electrons down a tube, and then the stuff it hits is not seen, and the electrons that go through are then captured on the other side. Allow much smaller things to be seen than with these next two microscopes. What you see here is a traditional light microscope, which rather than electrons, uses photons to bounce off a specimen to then hit back into your eye so you can see what you're looking at. Unfortunately, these can't magnify more than 1,000 times, but unlike the electron microscopes, they do not have to coat their specimen in metal, thereby not needing to kill their specimen. That is why, with electron microscopes, you cannot observe cells, but with these, you can. Thus, it was with light microscopes that in the year 1665, Robert Hooke saw the first cells, which he coined. And then, from then on, scientists started to develop cell theory, which, in the mid-1800s, was corroborated, saying that all living things are made up of cells. Now, have you ever wondered why there are no cells as elephants? Why you can see very few cells with your naked eye? Well, the only cells you can see with your naked eye are very large eggs, like chicken eggs, like the one you ate for breakfast today. However, kind of like this. Take this ram. Pretend this ram was a standard cell size, about 1 nanometer long. So, think about this. This ram has to get all its nutrients in through its outside surface area, right? It also has to pass through its membrane. So, if it wants some ATP that it wants to import from another cell, it has to come into the cell, into the through the ram's plasma membrane, and into its cytoplasm, so it can get used by the cell. However, if this ram were really big, say the size of this pillow, then the pillow really fluffed out. See, this pillow now has a very great volume, right? But it doesn't have that much surface area, so it can't bring in that much stuff to use because, as we know, all cells have to bring in, for example, oxygen, all things through their surface area. There's nothing to spoon feed it through. So that is why there are no huge cells, and why there are no tiny cells either, like smaller than like 0.05 micrometers, is because cells have to have stuff going on inside of them, and if they were too small, then they would be unable to have organelles and nuclei and DNA. Never mind the part I said about nuclei. And now, to talk about the difference between prokaryotic and eukaryotic cells, we bring in our senior researcher from our science department at the Felix Factor, Felix Heron. Give it up. Thank you. Now, to bring you an incredibly sensational analogy. Consider these two pencils. This pencil here is a prokaryotic cell. This pencil, a eukaryotic cell. Now, the chief differences between prokaryotic and eukaryotic cells are their simplicities. Now, take this eukaryotic cell right here. Of organelles, not many to speak of. Of organelles, many. There. Simply put, eukaryotic cells are much more complex. They have organelles such as the nucleus, the endoplasmic reticulum, the Golgi apparatus. So many different organelles. They can all do cool stuff. Whereas this dude, hey, he's just got regular, yellow coating, not even a cool tip, and like an eraser that's already faded out. This guy, on the other hand, so much cool stuff. Like, watch. Whoa. You got lead coming out of that thing? That's pretty sick. Got an eraser that you can pull out, get it even longer. Hey, you can even put in more lead. After you run out of lead, you can open this thing, put in more. Plus, look at that blue color. That's some beautiful blue right there. So that you can see how these pencils relate to two different types of cells. Thank you, Felix. So to sum it up, basically, ribosomes are present in both types of cells. These are the little protein-making little ball things that float around and stuff. However, in the prokaryotic cells, they have nothing more to speak of. Even their DNA is just floating around in little clumps. They've got a nucleoid region where the DNA sits. That's it. That's like old-fashioned stuff. You got eukaryotic cells floating around in my body, all got nucleus, DNA, nice and protected. Same for you. So, division of labor in a cell with the organelles, the compartmentalization and such, is really a staple when you look at organized labor. For example, take a look at industrialization. That was so successful because they had division of labor into different units of the cell, of the factory. Here, we have different organelles, for example, like ribosomes, which make proteins, endoplasmic reticulum packages them up, the Golgi body sends them off. These are all important things and can be done a lot faster if you have different spots for it. Plus, you have things like the peroxisomes, which take care of toxic things, which, if you had that stuff just floating around everywhere, that cell would not exist for very long. So after all this, you might be wondering, why am I holding a fern? And also, are there any differences between plant and animal cells? Well, the answer is yes. First main difference is the central vacuole in plant cells, which is a little compartment. If you can see these cells here, there's a little compartment in the middle of each of one, which basically holds stuff. It's like a big storage container. Whereas in animal cells, as you can see, there is none there. Also, plant cells have cell walls, which make them rigid, like this. See? It takes a lot of force to bend this, because the rigid cell walls in between the cells to make them tough. Whereas animal cells, they're supposed to be floppy and able to move their jaws and stuff and swim around. That is why there are no cell walls in animals. One of the major differences between plant and animal cells is, as you can see, this fern here is photosynthesizing, taking energy from this light from its chloroplasts and making glucose out of that. Now, in all animal cells, you have mitochondria, which make ATP. However, they don't need chloroplasts because, like this fish, cannot photosynthesize. It is impossible because there are no chloroplasts. Whereas in here, in plant cells, the chloroplasts contribute to their greenness. So last we have four main groups of organelles that we have. First, we have the manufacturers, the makers, like the nucleus, Golgi apparatus, smooth and rough endoplasmic reticula, and ribosomes, that good stuff. Next, we have the breakdowners, the detoxifiers, the peroxisomes, and the lysosomes, not to mention vacuoles. Third, we have mitochondria and chloroplasts, those which make or reformulate energy. And lastly, we have these structural things like the cytoskeleton, the microtubules, the cell membrane, plasma membrane, etc., etc. Plasma membranes, very interesting. As you can see by this realistic clay figurine, we have here fatty acid tails made up of methylene. These, similarly to most fatty acids and other lipid products, are quite hydrophobic. In fact, if I put water on it, it would not mix. I'm not going to do that because you already know what's going to happen, but that's what would happen. Here at the top, we have a negatively charged and therefore attractive to water, otherwise known as hydrophilic, head. What happens in a cell membrane is this. In a phospholipid bilayer, we have many of these. Let me see if we can get a better view of these little heads and members. What they do is that you have the phosphos together, the lipids together, and then we have the phosphos at the other end. And let's say this is the inside of the cell over here, and this is the outside of the cell. We got the cytoplasm sticking to this. Got the outside of the extracellular interstitial fluid sticking to that. And then we got the lipids sticking together, and it forms a nice, great semi-permeable membrane. So that stuff like oxygen and carbon dioxide can pass through, but then toxic stuff you might not want inside cannot. This is a nucleus. The middle of the cell, this is where the DNA is stored. This is where the chromosomes are stored. In the chromosomes, we've got some special thing called chromatin, which is what basically makes up the chromosomes. Now, in the middle here, we have the nucleolus. Now, this is where the RNA synthesis happens. Got two kinds of RNA. First, we got rRNA, which is ribosomal RNA. This ends up being formed to make ribosomes. Then we have mRNA. mRNA directs the ribosomes to synthesize proteins which are needed. For example, if you want to, copy some DNA, if you want to do some cell division, want to, make some phosphos. This mRNA tells ribosomes, make me some phosphos, and ribosome says, some phosphos and some lipids from the smooth endoplasmic reticulum, and voila, you get a phospholipid bilayer unit, which can then be part of this plasma membrane. Yay! So you might be wondering about the ribosomes at this point, which would be perfectly understandable. I'm wondering about them too. At least I was before I learned about them. Here's how they work. Ribosomes are little balls. They look exactly like this, except they're a lot smaller. And they make proteins. mRNA basically goes up to this ribosome and says, make me a protein, and then it makes it. Now, there are two types of ribosomes. First, we have free-floating ribosomes, like this one. And then we have ribosomes that are stuck to the rough endoplasmic reticulum, like this one here. The reason these are called, the rough endoplasmic reticulum is called rough, is because the ribosome stuck to it, making it look studded. So, that's basically how these work. When a ribosome makes a protein, if it's a free-floating ribosome, like this one, then it will flow through the cytoplasm until it gets to where it needs to go. However, if it's a rough endoplasmic reticulum ribosome, then the rough endoplasmic reticulum will take it, send it along to the Golgi apparatus, which will then make it into a protein and send it off on its way. This is the endomembrane system. It consists of the organelles that are enclosed in membranes, which include the nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, and the plasma membrane, and they are, serviced by little vesicles, which transfer proteins and stuff. Now, I must admit, endoplasmic reticuli are very useful. Pictured is a wild, rough endoplasmic reticulum that the Felix Factor crew caught in the jungles of North Africa. Now, as you'll notice, there are lots of little dots on it, and these dots are ribosomes that are attached to the endoplasmic reticulum, which is very handy because they can produce proteins that can then be used by the ER to synthesize into real proteins. And one of the main functions of the endoplasmic reticulum, believe it or not, is to produce membrane phospholipids that go onto the plasma membrane of cells. Now, I will now explain to you the process of synthesis, how a endoplasmic reticulum makes a protein. Well, the first thing that happens, needless to say, is the polypeptide that is being used is synthesized by a ribosome that happens to be bound to that endoplasmic reticulum. Happens like that it enters into a pore, and then it kind of binds, adheres to the shape of the rough endoplasmic reticulum. Now, next, what happens is that a short chain of sugars binds to this polypeptide, making it a glycoprotein. Glyco, by the way, means sugar, and protein is protein, which means glycoprotein is a sugar protein. Just in case you were wondering. What happens next is that it is put into a transport vesicle so that it can then be shipped off to the Golgi apparatus to be packaged and sent to another part of the cell. In case you were wondering, this is not a Hebrew letter shin. It is, in fact, a smooth endoplasmic reticulum. Smooth endoplasmic reticuli are very useful if you like living. They produce things that are important to life, such as lipids and oils, and they help process alcohol, which means that if you have an alcohol tolerance, that may mean you have a lot of a smooth endoplasmic reticulum working for you to help that. Another interesting function of the smooth endoplasmic reticulum is in muscle cells, where it is modified to store calcium ions as the sarcoplasmic reticulum. This is the Golgi apparatus. And in case you were wondering if it is only called the Golgi apparatus in order that it be easy to memorize for 10th grade biology students, the answer is no. In fact, it was discovered by an Italian biologist whose name was Camillo Golgi. Anyhow, the Golgi apparatus is like a stack of lots of different flattened sacs. And for those of you who think it looks like the endoplasmic reticulum, it is not, because, first of all, the endoplasmic reticula are connected, whereas these sacs are not connected. And well, other than that, they are pretty similar. In fact, they're both sacs. They're both organelles. Let's just forget it, okay? Next, we see the function of the endoplasmic reticulum. You can tell that on one side is the receiving end. Vesicles with glycoproteins come into the Golgi apparatus, which then modifies the carbohydrates on the glycoproteins and other interesting molecular identification tagging, at which point it then ships it along through the regurgitation end of the Golgi apparatus to its next destination. Lysosome, the endomembranous sac which breaks stuff down. Did you know that in Portuguese, lysosome means digestion? Well, it doesn't. In Greek, lysosome means breakdown body, which is why lysozymes, which are little enzymes in tears, break things down like pathogens that are trying to enter the body, which is why those are called lysozymes. This is a lysozyme. As you can see, there's a membrane around it. In the middle is where stuff goes. Lysozymes serve two main purposes. One, to digest food particles. What happens? Food particles go into the lysozyme, and using its digestive enzymes, it breaks them down. Secondly, to break down non-functioning parts of the cell. For example, if you have a piece of an endoplasmic reticulum that was not working, it would go into the lysosome, and digestive enzymes at work, and out you have organic molecules, which means they contain carbon and are not CO2. This is a vacuole. Vacuoles serve so many different functions, they couldn't even list them all in the textbook. However, here are several. 1. In collusion with the lysosomes, they help bring over food to be digested by the lysosomes. 2. In protists, they help get rid of excess water in a lysosome. You get lysosome in the spokes, and then it out through the center of the thing, and the excess water goes out. 3. In plants, the central vacuole is like a big sac that contains water and helps maintain the general homeostasis of the cell. We've reached the halfway part of the show, so now I decide I'm going to tell you a science joke. Ready? A neutron walks into a bar and says, How much for a beer? The bartender says. For you, no charge. Ha ha ha. Now for a quick review of the manufacturing of proteins in cells. First, we have a polypeptide, which is synthesized by a ribosome, which, in this case, is attached to the rough endoplasmic reticulum. The rough endoplasmic reticulum makes it into a glycoprotein, along with a carbohydrate, which is a little short chain of sugar. Then, it is packaged into a vesicle, which off to the Golgi apparatus, where it's taken in by the Golgi apparatus, packaged up, and then sent along on its way in another vesicle. And then we got peroxisomes, which are not part of the endomembrane system, but they do apoptosis when they tell cells to do, and basically just explode stuff. Without a doubt, my favorite organelles. So there's going to be a twofer, guys, so watch, it's going to be pretty fast. Make sure you fasten your seatbelts. So we're talking about the differences and similarities between the energy processing in mitochondria and chloroplasts. First of all, chloroplasts came first, I believe. These were little singular prokaryotic organisms that got assimilated by the cells. Symbiotic relationship here. That's why they have cell membrane around them. True story. Also, they have their own circular DNA, which is like, and a little Mobius strip around them, which is pretty cool. And now some differences. Now, first of all, we've got in the mitochondria, we have all the energy processing and the ATP stuff that gets done in their cristae, which are the little folds in the outside of the mitochondria. Now, in the chloroplasts, we have little sacs, which are called styracoids. And inside of these, we've got the stroma, which is a thick fluid. Now, each stack in each thyroid, that's called a granum, and these are the things that make the sunlight into the glucoses. Oh, also, the mitochondria has the intermembrane space, which is where and also we have the mitochondrial matrix, which that's where the DNA is located. Needless to say, there is plenty of evidence to support this. Take, for example, this mitochondrion. Now look at it. Look at that plasma double membrane out there. When you look at that inner membrane, you just think, why? That looks just like a prokaryote membrane. Also, look, it has ribosomes. They're invisible in this one, but they're there. Also, it has DNA, circular DNA, just like many prokaryotes do. This is pretty hard evidence that mitochondria and chloroplasts, which are very similar, both involved by endosymbiosis. Still not convinced? How about this? Look, when this mitochondrion is going to reproduce, that was just like prokaryotic reproduction. I'm telling you, hard facts, you can't dispute it. This is a microfilament. You might think, because of my exquisite drawing, that it is actually a DNA strand, but it is not. It is a microfilament. It is made of axiom. It's a solid rod structure, and its function is in contraction. It interacts with a motor protein myosin, which contracts, and it generally just helps hold cells together. Now this, despite my best efforts, is an intermediate filament latching a Golgi apparatus into place. Now, this might look a little bit strange, but in fact, this is exactly what intermediate filaments look like. They are rope-like structures that are bound tightly together from proteins, and their main function is, besides supporting the cell, to attach organelles. Now this, as you can probably deduce from my exquisite penmanship, is a microtubule. Now, things about microtubules, they are hollow tube things that they are the most strong, and they are used in cilia and flagella. They flap around and stuff, and they also make up centrioles. And along with extending from the basal body, their main function is to flap around and to move the cell. This here is a cross-section of a flagellum, drawn masterfully by yours truly. Yes, is this the MFA? Yeah, this is Felix. Want to put it in your modern art collection? Let's settle the figures later. I'm sorry, I'm in the middle of a science video. Sorry. Anywho, this is, you'll notice, made up of a 9 plus 2 arrangement. There are 9 doublets of microtubules around the outside with 2 in the middle. And that is the way these are made up. Now, also, in between each doublet, not pictured, simply because El Maestro ran out of the right type of color for his painting on Magnifico. There are little diamond arms which grab onto the other side of the thing to make it bend. Now this funky thing is called a basal body. This is basically the centriole for flagella and for cilia. As you'll notice, it's a 9 plus 0 arrangement with 9 triplets of tubules on the outside and nothing in the middle, and it's out of these which the flagella and the cilia grow. The matrix. Probably not the same kind of matrix that you were expecting. However, this matrix, 100% of biology teachers assure us is more interesting and more fun. Here's how it works. This matrix is made up of collagen glycoproteins. Glycoproteins being proteins with small strands of carbohydrates on them, attached to integrins, which are membrane proteins, inside between two phospholipids. Basically sticks out, attaches to a connecting glycoprotein, which then you got these tough collagen fibers which are outside the cell. Basically, this helps to support the stuff out there, connect cells and stuff, make sure they don't, just fly around. Also, excellular matrix can be sticky, which helps cells to stick together. Junctions. Spots between cells. We have 3 main types: tight junctions, gap junctions, anchoring junctions. They're all very useful. Now we're going to find out about them. By the way, I ate a blue lollipop, which is why my teeth and my lips are blue, so that's not actually weird. Just, don't mention it, please. Similarly to how caulk works in bathtubs by creating a waterproof seal by sticking a little bit of a waterproof substance in between two things. For example, in this picture, the bathtub and the tiles on the outside. A tight junction does the same thing for two cells that are next to each other, adjacent. What it does is there are little lines, little rivulets that are full of waterproof substance. Basically makes those two cells impenetrable to water. Pretty useful. This is an envelope with a little metal clasp on it. How this relates to cell junctions, I will tell you presently. You see the little clasp thing? Well, picture that and another cell. Picture those two clasps digging in and then pushing back out, like on the top of the envelope, like when it's closed, like right now. These are just like cells, except at that top layer of the envelope, clamping them together. So now our last junctions are gap junctions. This analogy is a little bit crazy, but think of this orange you see here. Think of all those little pieces of orange as separate cells. A gap junction, or the little tubes connecting them, allow the quick relay of ions between them. For example, in your heart, you can get quick transfer and to make sure your heart beats all at the same time through the junctions. Like this one. Remember when we studied the heart and we said it had junctions? Well, these are they! So that's basically how it works, which is pretty neat. Look closely now. You don't get to see art like this every day. Pictured is a plant cell wall in its magnificent splendor. In the inside, you can see the two primary cell walls, which are made by the plant to be sort of flexible so we could keep growing. However, on the outside, you see the rigider secondary cell walls, which are tough, and they are made of cellulose, and they are what keeps the plant nice and sturdy and straight. Also, you can see the two little holes in between the membranes of the cells. These are called plasmodesmata. These are the gap junctions, except in plants. And if you cannot remember what gap junctions do and how they function, please refer back to chapter 4.21. After all this, we should be pretty familiar with all the different organelles you might find in any old cell. But we can make this even easier if you can't remember, grouping them into 4 different categories. First, manufacturing. You can probably guess what's in there. We've got the nucleus, which does RNA synthesis. We've got the ribosomes, which do protein synthesis. We've got rough ER, which makes the it's basically like finalizes the proteins, and we've got it also makes transport vesicles. Can't forget transport vesicles. And we've got the smooth ER that does lipids. It also detoxifies. Lastly, we've got the Golgi apparatus, which modifies the macromolecules made in the rough ER and ships them off in transport vesicles. Next, number 2, we've got the breakdowns. Here we have lysosomes, which, as you remember, break down food and also help to break down dying cell parts like organelles. Then we have the vacuoles, which digest stuff. And also we have peroxisomes, which are not part of the endomemory system. However, they release carbon peroxide after breaking down diverse molecules. Third, we have energy processing, which only involves 2 things, both of which are very similar, however different. One of them is the mitochondria, which are the powerhouses. They synthesize ATP from carbohydrates from plants. And we have the chloroplasts. And these chloroplasts, their job is to create glucose from energy from the sun. Fourth, and lastly, we have the support, movement, and communication. That includes the cytoskeleton, which of course includes the microfilaments, microtubules, and intermediate filaments, basically holding up the cell, making sure it runs. Also, microtubules are used to create cilia and flagella. Also, we have the extracellular matrix, which of course is made of glycoproteins, make sure the cells stick together well, and yeah. And lastly, we have the cell junctions. These are good for communication between cells, making sure they stick together nice and well. And very lastly, we have the cell walls, which in plants and fungi and some protists help to strengthen the cell and make it strong. Take me out to a membrane, take me into a cell. I want to see all the organelles. How many ribosomes there are, I never will tell. There are 3 characteristics of organisms. You'll find them in every one. Regulated cells, DNA, and metabolic use are in every cell. Each cell has a membrane which maintains internal conditions. In cells, DNA is the molecule that tells organelles exactly what to do in metabolism. Materials are taken in and then used on the inside, and then soon thereafter the wastes are excreted out back to the outside. Take me out to a membrane, take me into a cell. I want to see all the organelles. How many ribosomes there are, I never will tell. There are 3 characteristics of organisms. You'll find them in every one. Regulated cells, DNA, and metabolic use are in every cell. Regulated cells, DNA, and metabolic use are in every cell.